So good morning to everyone. It's a great pleasure for me to introduce Raphael Aguillon, who's the first year postdoc fellow who comes to us from France, where he did his PhD in the University of Toulouse. And Raphael is doing something unusual because he's doing his postdoc in two different labs at Bar-Ilan University, in the lab of Oren Levy, I believe, and Lior Appelbaum. That's unusual. And he's going to speak about something which is, I think, certainly not overrated. People think of people's needs, and they think of different needs, but obviously sleeping is the most important activity. We all know we waste about a third of our life on sleeping. So Raphael will speak about a very interesting question: which came first, sleep or the brain? So thank you.
Thanks, Elliot. So as you just said, we spend 30%, a third of our life to sleep. So if we come back to the time of the day, it's like in average 7 hours. And if you compare to eating is 1 hour, 1 hour and a half. So it depends on the people and the culture, but it's on average. And yet our society, we are obsessed with food. And if we consider the timing we spend eating compared to sleep, it's kind of very strange how much we don't focus so much on sleeping. Just think about 1 minute every day, okay, at the same time you go to bed, you close your eyes and boom, 7 hours later, you woke up and you are ready for another day. It's like, what just happened? A third of your day just vanished and we don't know why yet. So you tell me that, yeah, but Raphael, when I don't sleep enough, I feel so bad, my mood is bad, mine is very bad, just 1 hour less, it's awful. Just ask all the people of the lab. My attention, my memory, everything is affected. All of these are consequences. In fact, there is no consensus in science, in neurobiology, of the why do we sleep. And the question is, how can we nail it? How can we discover it? So as a biologist, what we do when we have such a question, we unfocus from human and we start to explore all the other organisms. And we start to explore sleep to try to identify it. So first question, like what is sleep for a scientist? Like how can we see it? So there is two tools, let's say. First is it's a very specific behavior, okay, with specific criteria. I will come back a bit later after. And it has as well a specific brain signature. So we looked for it over decades and we found it in the most different animals, ranging from mammals to insects. And if you look at the quality of sleep of these animals, there is no coherence. It's very puzzling for scientists because some animals are sleeping during the day, some animals are sleeping during the night, some it during hours, and some it's like few minutes. Sometimes they don't move, sometimes they swim, sometimes it's half of the brain. It's just nonsense from outside. The only things we can say for now is like, okay, if you have a nervous system, if you have a huge brain or a very small one, because insects have a very small brain, but they do have, you need to sleep. So it's something which, of course, and it's basic and simple, it's happening inside the nervous system. And recently, we had like newcomer in the game of the sleep science, these guys. So for many, people don't know so much, but they are animals. We call them Nidarian. It's a group of animals, very strange. When you look at them, you don't even think it's part of the same planet. And they do move, okay? They move, they have like nervous system too, as well. So in this group of animals, of course, you know the jellyfish if you go to the beach, and as well, if you dive, you know the corals, it exists as well other animals, which are the sea anemone. Okay, so it brings, like, it's very recent. It's work from the last decades, and we discover, and it's a hot place for a neurobiologist of sleep to be, because everything is new. We try to describe it in different animals. We don't know. This is a place to be, and it adds this new value in the global picture. What do they all have in common? So they do all have in common a nervous system again. But in fact, apparently, from this last result, you don't need a brain to sleep, okay? Which gives us the last option, the neurons, which is a unit of the brain, okay? And I do think that we can find the why of sleeping by investigating sleep in such animal. And during my project, I focus actually on one of these animals. I don't know if you see. Wait, laser point. Okay. Me, I focus on one of the animal of the sea anemone. So mine, his name is Nematostella vectensis. Here it's a sketch, it's a small one, okay? It's 1-2 centimeters. It lives during the night in the mud, it hides. And first question is how and when do they sleep? Look at it. I mean, like when I start my project, I was like, okay, let's look at it, and maybe it will do something, and I will understand. And I spend hours like this, and okay, it move. Oh, it stopped to move. No, maybe I don't know. So you can't guess like this, and this is what brings me to explain you the definition of what is sleep. Okay, sleep in terms of behavior has a very specific criteria. Okay, so if you follow the animal during like 24 hours, okay, you follow the movement, it will be a period of time where the animal moves less, okay? And during this period of time where the animal moves less, the animal has to be less sensitive. You have to stimulate more to make him react. Another point, it has to happen roughly at the same time of the day, every day.
Bonjour, comment puis-je vous être utile?
Ah. And the last criteria, which is very important, it's inescapable. It means that if you stimulate, or if the scientists stimulate an animal while it is supposed to sleep, so it wake up, at the end of stimulation, they will fall asleep. There is accumulation of a need for sleep. You can't escape from it. So in order to describe it, for now, I collaborate with a brilliant student, Amir Ardouf. So we are both together exploring and using different quantitative tools in order to detect the fine parameters of when and how the animal is sleeping in a very simple setup, as you can see here, in order then to go to a more ecological setup. So let's come back now to my fascination to neurons. So in the lab, we have a very specific nematostella, which has been genetically modified. They have been engineered. And this modification makes the whole system nervous fluorescent. It's amazing because look at the animal on the picture. If you look at it, you don't see anything. And when you want to see neurons, you have to make it visible. And this is one way to do it. So while the animal is alive, you just use a very specific set of light. And suddenly, because the animal is translucent, you see all the neurons which appear. Like you can see here, it's a close view on just 2 tentacles. And you can see the filament, which are the body of the neurons here, okay? And basically what you see here, it's the most complex part of the nervous system. I told you, there is no complexity. It's just a mesh of neurons connecting one to the other one, okay? And that's it. So for me, as a biologist of cell biologist of neurobiology, like now I have a way to zoom in with powerful microscope and to dive into the neurons while the animal is sleeping, and I can start to ask questions. What's happening while the animal is sleeping? So, in fact, I work in a lab in your Applebaum. We investigate the peculiar and amazing process. So we focus mostly in the lab, focus on the fish. And what we know, so in neurons, during the wakefulness, so neurons are contacting, are sending information to other neurons, okay, to send like very relevant information. This information travel very fast. It's like a powerful wave of electricity, let's call it like this. And it's so powerful, it has consequences, okay? And these consequences are damages. Because of the normal activity of the neurons, it has damages. And these damages are, we know some of them are DNA damage. So DNA is it's very important, of course, you might know it's what makes your organism working properly and have a correct physiology. If it breaks, you don't want to keep it break long, okay? So we know that during the day and wakefulness of few animals, you have accumulation during the day in neurons, okay, of DNA damage. It break. And then the animal go to sleep. You have the electrical activity of the neurons, which shut down, okay? And then you have all the cells which work on repairing all the DNA damage to be ready for the next day. And in the lab, we do think that this is why we sleep. And in order to know if this function and this process exists since the beginning of the nervous system, I do investigate this question in the small sea anemone that I told you. We do think that in fact sleep exists since the first neuron, since it exists. And without sleep, I think we think that there would not be any nervous system. So I want now to conclude, to come to conclusion, to unfocus a bit. So we were very deep inside the neurons. Okay, this is where is my daily life actually. And I want to come back a bit on the size of the animal, and then to come back to more, for me, what is the main information of this talk of today is we all sleep, okay? And individually, you all experience the effect of sleep deprivation for many reasons, because you are parents, because you have a cat, because your neighbors are doing parties, or you're having parties, or maybe because you are living in Tel Aviv downtown, and you know what I'm talking about. So you might, and you know how bad it is to not sleep enough. And if we all sleep, for sure, and there is no question about it, they all might suffer from it. So I choose this nice picture. It's a beautiful one from Brazil. And I could choose many, many other cities. But this one is very interesting because you can see something very interesting and how much I would say the city overlap with the nature. Okay, you can see like the city and it's during the night, all the lights, and you can see that there is a sea, there is a mountain, there is a forest, and you have on this us. So I think now it's time to ask this question, and I want to give it the question as a takeaway. This is for me my big question when I am in my sea anemone working. I have this huge question, and I really want to work on that. It's, does human activity cause global sleep deprivation? I wish you a very good day and a very good night, and I'm ready to take any question.
I have one question, Raphael.
Yeah.
So, so one of the bullet points in your definition of sleep was this notion of daily, right?
Yeah.
And I guess this has to do with the sun. So are there like other like other types of sleeps? Like for example, how do deep sea creatures sleep, where there is like no notion of day and night? Does that make sense?
Yeah, it makes sense. You ask me, let's say, like for the animals which are not connected to
the solar cycle, yeah, the daily cycle. Yeah.
They do sleep. Yes, it does exist. They don't need to see the sun to sleep. In fact, if you even if you take any animals which live under the sun, like us, and you put like in a constant darkness during days, the animal will still sleep, and at the same time of the day, even if it doesn't see the sun, because we have inside what we call the biological clock, and the sleep is like controlled by it. Okay, you need to sleep, but the clock is telling you when you sleep, so you don't need to see. Okay.
No, but this is like, I think you're taking a human and putting him outside, right? I'm talking about, like, for example, deep sea creatures. They don't have a notion of day and night, I guess, right?
Yeah.
And this applies to them too?
As I told you, in fact, like, okay, I understand what you said. So for what I know and for what we know, sleep is, it's an independent process, and it's using different inputs to know when it happened. Okay, it can use the light, but it can, for some animals which are not using or cannot see the light, you can have as well the tide, and as well it can be the moon, but the moon will be related to the side. But it's not like it's independent. Okay, you have like animals using different cues to know at which time of the day they are. Okay, for the deep sea creature, it's not well known, but they do have a clock, and we don't know yet exactly how they get information.
I see. Thanks.
Sure.
Raphael, I have another question. Why did you exactly choose this kind of animal to study the sleep and all the process of sleeping, and not, for instance, other type of animals?
Yeah, so I was looking at the end of my PhD for an animal which has one neuron. You know, I'm this kind of biologist, and I'm like, okay, if I take an animal with one neuron, I will make the link between the behavior and the activity of the cells. In fact, I'm a cellular biologist. I want to understand the world from the cell. And I didn't find it. But what I identify is, and I understood, is like the most primitive nervous system which exists is, in fact, inside the nidarian. So I joined this lab, and they have like few of them. And this one is very convenient because it's very small. It's not like a huge jellyfish. And like, I don't want to get burned all the day as well. So yeah, this one makes sense for different reasons. But the main one is the fact it has a very simple nervous system. It has no, let's say, as far as I know, no cognitive function, no, I mean, like all this sleep deprivation effect, the mood effect, all of this hide a bit the function of sleep. And I was looking for something when I don't have so much process which hide the truth of why we do sleep.
Raphael, there are a few other people who have raised their hands. I see Jan and Andre, and I saw before Belshazzar and Natalie, I think.
Okay.
So can you answer very briefly? We have a few more minutes.
Okay.
Yes, thank you for the talk. So it's very beautiful. You can color the neuron and then see them. But is it useful for the research, for your research, the fact that you can see them?
Yeah, so it's in fact the base of my work, because what I'm asking is what's happening inside the neurons. So I need to just to locate them. Without this system, I can't see them. You know, the animal is transparent. So if I put a nematostella in the microscope, I see nothing.
I see.
Okay. If I have a transgenic like this specific animal, suddenly I will see nothing except the neurons. And then I can use specific protocol to understand what's happening inside.
I see.
Okay, to make it visible.
Can I ask?
Mm-hmm.
So it's a two-parter. First, if I understand correctly, you identified in this organism that the reason for sleep is kind of the damage that the neurons do to themselves in their normal activity. So how hard or how far away you are from or someone else would be to show this for humans? And the second part is, would there be, once you know the reason, is there like some sort of practical application? Could you come up with a way to maybe shorten the amount of sleep or improve the quality of sleep or something like that?
Yeah, so for human, it's like a tough question to exactly show it like this, because you need to access to inside the neuron while the animal is alive. So it's not very ethical. So, but we are looking for, let's say, molecules and markers which are released by this system, which can give us some information that it's still happening. So in fact, what we know, if you take the blood of humans who are sleep deprived, you can detect DNA damage more than usual. So apparently it's something which might happen in humans, but it's difficult to know it, to explore like in animals. For the second part of the question, it was, what was it? Like, what are the potential application? So first, like prevention of sleep, we don't need so much science for that, okay? We know exactly how to sleep good, but it exists like different pathology, different contexts where like it's a problem and the prevention is not enough. So like recently in the lab, we discovered that there is a specific molecule. I don't know yet if it's a very good, if it will be like a drug by itself, but it's these drugs, it like makes the cells not seeing the DNA damage, and the animal don't feel the need for sleep. So at the end, you know, this kind of things, I'm like, oh, it's amazing. So maybe I can work more. But at the end, if you don't feel it that you have DNA damage, you don't go to sleep, you don't repair. At the end, it's a problem. You mean like to have DNA damage, it's a normal consequence, and we have the whole system which knows how to deal with. In fact, it tells us when you need to go to rest. So I don't know how, if at the end, like the output of all of this will be to design drugs to sleep less. I can't tell you. I'm not sure this is where we go, but you know, who knows?
Thanks.
Andre?
Yeah, okay. Thank you for the presentation. I have one, maybe two stupid questions. So as far as I understood, you observed the neurons in real time using an optical microscope, right?
Confocal.
Confocal microscope. Okay, and you look at these neurons in real time, but the animal is constantly moving, I guess. So it can be pretty hard to stabilize the picture.
Yeah. So the question is how I do that.
Yeah. Right.
Okay, I lie to you. No, in fact, what I'm doing, so I have two approaches. For now, what I do, I take the animal this is why I detect, I try to understand when the animal is sleeping and when he's supposed to sleep. I take a batch of animals and then I look inside. So an animal cannot behave, but I know at this time of the day, it's supposed to move less. Okay, for now, this is the main approach I have because indeed, like the animal move too much. But we are starting collaboration in Germany with like crazy people about microscope, and they are generating approaches to generate microscopy, which allow us to detect cellular signal while the animal is behaving. So it's like under construction. This.
So there is quite complex engineering construction.
Yeah.
Maybe it's some kind of optical slicing.
Yeah, it will be like very fast, probably like, I don't know if you know spinning disk and this kind of technology of confocal. It's like even faster than that, and but I don't know yet. It's a collaboration we are going to start and
okay.
But yes, the point is to indeed detect like in live in the cell while the animal is moving, which is a challenge technically speaking. It's very difficult.
Okay, thanks.
Sure.
We have one with time for one more question. Flavio, I see you raise your hand.
Hey, Raphael, wonderful presentation, really nice. So, you know, I work with meditation practices, right? And it's all about relaxation in general, right?
Yeah.
And so I wish I could I could teach these little animals to meditate. That would be a lot of fun. To figure some stuff out. But I'm wondering, I mean, it's remarkable that you go to the most simple form in a way of being that you can actually see these neurons move. So I'm wondering whether we could use this do people use this to test other stuff, like how to deal with trauma, with stress, if you put them together, if you put them some kind of drugs in there? I have no idea, right? Like, is there experimentation beyond sleep going on these? Because in a way they're connected, right?
Yeah, of course. Like I know that in my former institute, they used to work on PTSD on mice, and they used to detect neuronal activity and other neuronal process while the animal is experiencing the PTSD, the memory when it flashback. Okay, so yes, it's a very active field of research.
Could you keep them awake for a long time? Like, can you keep poking them?
Yeah, like any animal, of course. If you manage to know how they sleep and how to wake them up, you can annoy them. This is what I'm doing with my sea anemone. I just poke them regularly to annoy them. This is my job.
So what happens if you keep poking it? Like if you don't let it sleep?
Okay. You want to know? Okay. So sleep deprivation at some point lead to death after days of sleep deprivation. We know that. So yeah, this is a consequence. Thank you.
Thank you, Raphael.